Welcome everybody to today's webinar presented to you by Biognosis. Today we're going to talk about monitoring the performance of a proteomics core facility, troubleshooting using our quality control software QuiC. My name is Florian Marti. I'm Director Global Product Support of Biognosis. Today we're going to introduce you QC analysis for proteomics, what our best practices are. Then we're going to look at some new features of QuiC 2.0. And then we will have a guest presentation by Joanna Kirkpatrick, manager of the core facility for proteomics, Leibniz Institute on Aging. And after that, we'll follow with some question and answers from you that you can address to either Joanna, me, or my colleague Ian Leonard, who's also sitting with me here today. Quality control, or also called QC, is an elemental process of quality assurance in proteomics experiments. QC metrics in generally can provide important information on sample preparation, liquid chromatography, and mass spectrometric acquisition, and are important to determine the sample quality and judge whether samples can actually be excluded from data analysis or whether your entire LC-MS setup is performing well. In generally, quality control should be done on the sample preparation as mentioned, on a mass spectrometer, on the spectra, but also on the back end, the bioinformatics part of one experiment. In general, QC analysis can be done either inter-experiment or intra-experiment. When doing inter-experiment QC, the user is mainly interested in the LC-MS performance over time as depicted down here. When doing a large-scale experiment over time, we want to measure regular QC samples in between our samples of the experiment to judge the LC-MS performance, to in the end tell our customers or the users of the LC-MS facility that everything with the mass spectrometer was fine and the data can be used for data analysis and biological interpretation. So in general, in inter-experiment QC, we want to monitor the trends of the LC-MS and perform maintenance before something breaks down and it is too late and we lose important mass spectrometry time, or even in the worst case can actually lose even samples. On the other hand, intra-experimental QC is done, as the name says, within one experiment, and here we want to actually monitor the performance of the LC-MS within one experiment, but we also want to assess the sample quality of different samples for the entire study. As depicted on the cartoon here, again, we're measuring over time, and now we want to see if all of our samples are actually behaving within boundaries which we consider acceptable for sample quality, for mass spec performance, but also for number of IDs that we get. On the basis of intra-experimental QC analysis, we can then actually exclude bad runs on the basis of objective measurements, and this helps us to save valuable time on the data analysis. With QuiC 2.0, also called Senna, we launched recently an easy to set up, fast and intuitive QC analysis software on the basis of the previously launched QuiC 1.0. QuiC 2.0 supports major proteomics workflows such as DDA, DIA, MRM, and PRM. To our knowledge, QuiC 2.0 is the only QC analysis software out there that actually has a proper handling of corrupted files. One of the key new features about QuiC 2.0 is customizable reporting for documentation of the performance of the LC-MS facility. Furthermore, the QC measurements can sort by acquisition date or other measurements. On top of that, we have added completely automated DDA performance monitoring that don't need any external search engine. Furthermore, we have updated all the most of the plots in QuiC 2.0 to make them more intuitive and more informative. And one of the most recent features that we're still finishing up is actually very soon we're also going to launch a full compatibility with MStats QC, so that the output of QuiC can actually be loaded into MStats QC and used for further analysis in there. As I mentioned at the beginning, so some of the new features are as depicted here, more intuitive plots, for example, for the MS1 mass accuracy, but also added new plots, for example, for the IRT accuracy within one run, where we even describe the R-square between the measured IRT and the retention time to give an indication about how stable the chromatography was within one run. Then what we added as well is event tags that can be programmed in the file run name and indicate at what time of the QC analysis a certain event has taken place. As you can see here, for example, we've actually coded in the name different modifications, such as exchange of the transfer capillary or recalibration, adding new columns, and these are then showing up in the QC analysis and also later in the QC report, so that we can actually show this to our customers and show them when we did some interventions at the LCMS. This is it about the short introduction about QuiC 2.0. I will now hand over the microphone to Joanna Kirkpatrick, and she will actually describe us how she does QC analysis using QuiC 2.0.
So hopefully you can hear me, Florian?
Yes, I can hear you.
Okay, good. Then I just start my presentation mode, hopefully. Okay, can everyone see my screen, I hope? So yeah, thanks for the invitation to give this presentation, Florian. Yeah, my name is Joanna Kirkpatrick, and I run a core facility proteomics at a Leibniz Institute in Jena, in Germany. So yeah, just a quick overview of what we do here. Our institute is a research institute situated in Jena, in East Germany. It's an international university city right in the middle of Thuringia, and we aim broadly to research at understanding the molecular mechanisms of aging. And within the institute, we have various different animal models that are used, mice, different kinds of fish, worms and flies that help us generate relevant knowledge on the causes and the mechanism of age-associated diseases. And we have scientists from over 30 different countries working here, so it's quite an international institute. And I've been on a core facility now for nearly 2 years, and we provide a full support from the project design to data analysis for scientists, PhD students. And we offer a complete platform for identification and quantification of proteins using mass spectrometry. And since we started the facility nearly 2 years ago, we're now running projects across, actually that's pretty much all of our internal and associated research groups, 16 external collaborators, and we have a total of 66 projects that have been running since February 2016. So we have a very broad spectrum of users for which we obviously try and keep our services working all of the time. So in our facility, we now have 2 instruments. So since the end of December 2015, we had a Orbitrap Fusion Lumos installed, coupled with a Waters Nano Acuity. And just this year, a couple of months ago, we got the brand new QuiC Active HFX, also coupled with a Waters Nano Acuity. And these instruments are now both running 24 hours a day, 7 days a week in the facility for our users. And so we also have different kind of users. So I have people I call my core users. These are regular users of our proteomics services. So we have some research groups here who are really focused on proteomics as their major research tool for investigating their research questions. So for example, we work very closely with the group of Alessandro Ori, and together we develop and establish new pipelines and tools, which we then offer to our other users. And then we have facility users. So the other groups that come in, their samples come via us in the facility, and we handle all of the experimental design, sample preparation, data acquisition, and processing. And these are users for whom perhaps the proteomics is a smaller part of their project or an occasional project rather than the main focus of their entire research project. So what do I do in terms of monitoring the instrument performance? So first of all, I tell you a little bit about my QC sample and why I use it. So when we get our new instruments, of course, we evaluate the performance of those in the first instance using the Pierce HeLa protein digest standard that one can buy from Thermo. And this is a very good and reliable standard to use to benchmark the performance of the instruments when they're installed, but it doesn't come cheap. So against this performance, I then run my own QC sample, which is an in-house digest of MEF, which is mouse embryonic fibroblasts, which I can generate in very large quantities reproducibly. So I have a technician who fortunately does a very good job in cell culture and prepares me many batches of these 10 million cells, which we keep stored in our -80. And when I want to prepare a new batch of MEF, I digest these 10 million cells. This gives me around 1 mg of protein digest, which I can then store in aliquots and bring it out as and when I run out in the instrument. And I add the HRM kit in order to be able to look at all this data in QuiC, just before I put the new samples into the autosampler in my instrument. So what do I do on a regular basis? So I load 1 microgram on column for my post-calibration weekly QC runs in both DDA and DIA. And also, as Florian mentioned, all of my users, they fully QC their whole sample preparation process as well with a smaller number of cells from the same cell culture experiment. We also have many batches of those in our stock, so everyone processes one of those alongside their samples so that they can also quality control what they did against the performance of the standard, which is prepared on a large scale. And the really nice thing is then that I monitor DDA performance with 1-hour runs during beginning and end of every batch, and also sometimes in the middle, depending on the length of the batch, and always post-calibration. And the DDA is also used to then evaluate the user-generated sample. And my DIA performance, I currently monitor it with the longer gradients. And this is really only possible to do with QuiC. Before QuiC was there, I could only really use the other tools that I was previously using for QC. I could only really do that with the DDA. So that's the really nice thing about the QuiC as well. So what do I monitor? Of course, MS1 intensity is a really obvious and easy indicator that things might not be quite right with the instrument. Looking at the mass accuracy, both in MS1 and MS2, how many proteins I identify from my sample, and also from those protein identifications, actually how many peptide and precursor identifications I get. And before I had the QuiC software, I was obviously just looking at raw data in Excalibur to look at the MS1 intensity. This obviously I still do. And then the rest of the information I was extracting by searching the data in Proteome Discoverer using Mascot for all of the other parameters. And whilst this is a good tool to get the necessary information for DDA, it requires a little bit of effort on the part of the person who's actually doing it. And whilst I do it, it's something that it's not always so easy to get the regular users to do when they're checking their own QC sample and the before and after quality control sample that I've prepared. It's sometimes a little bit challenging to persuade people to invest the time to go through the process of doing this. And I look at this in a bit more detail so you can see that perhaps, yeah, why people were a little bit reluctant, because it's a little bit time consuming. So first of all, you have to open your study in Proteome Discoverer, transfer the files from the server, select the QC workflow and send the search, and then you have to wait. And then you have to also wait while the resulting consensus file opens. So this is just an example of a short run of my MEF and the kind of information you get when you open the consensus from Proteome Discoverer. So you see at the bottom, and I highlight it, so hopefully it's a bit easier to read because it's quite small on the screen. You get the information about how many proteins, how many peptides, how many peptide spectral matches, and then how many MS spectra were acquired during that run. And of course, then one can evaluate identification rates and other things quite easily by making calculations on these numbers. However, it is a bit manually time consuming. If you want to monitor any other parameters, you need to open another viewer to look at the different parameters like mass accuracy. And to check whether the performance is consistent over time, you either have to go and open old searches for comparison, unless you've got it in your head. So apart from me, no one else in the lab remembers what happened last week. So it tends to be a bit time consuming to go and look and open an old file just to check that the performance is the same as it was at the end of the last person's batch. So for example, yeah, there's all these plotting tools within Proteome Discoverer, so one can easily bring up another visualization to look at the mass accuracy, for example. But again, it's more clicking, more effort, and people are kind of inherently a bit reluctant to invest the time in this. So this is then how I've been using the QuiC workflows since I started using QuiC 1.0 and now QuiC 2.0. So what I really like is that I can upload the data directly from the folder where my data gets pushed on our server, so I've got no need to copy it. I've created libraries using either Pulsar or MaxQuant from a batch of my QC runs in DDA, and then these are really easy to save as a reference library for the individual instruments and also for different runtimes if you need. And then you can just select whichever reference analysis you want for whichever kind of data. Each type of data is recognized by the software automatically. I really like this because then I don't have to decide where it goes. It knows whether it's DDA data, it knows whether it's DIA data, it knows which instrument it came from, so I've no need to shuffle files around afterwards. And it's really easy to monitor the performance using primarily the precursor and protein identification numbers, but as well as the other parameters. So just to give you an idea, this is sort of how, for the instrument I've had longest in the facility, this is for my Orbitrap Fusion Lumos, how the file structure looks with the data as it gets automatically loaded into the software. So this is nothing that I've had to do. This is how it does it itself, and it means it's really, really nice. I can go back and find anything I need from whichever date. It tells me not only when I acquire the data, but when I uploaded it to QuiC, so I can see if perhaps I did something silly at any point in time. And then this is probably my favorite output and the one I use most often because it's such a quick and easy way to see exactly what's going on. So in the green line, you see how many precursors I have identified over time, and on the bottom, the red line is my identified protein groups. And what I'm going to do now is give you a couple of examples of how I then use the QuiC to troubleshoot these dips in performance. So there's a couple of dips you see in performance at the top. One back in early February this year. This was actually due to a rather inefficient cleaning from the engineer who was a bit impatient and didn't do a particularly good job, so I had to re-clean the instrument. But the one later in the year, just at the end of August, this was actually a bit more challenging, but the QuiC was really useful in order to be able to work out what had gone wrong here. So what I really like is that, it's really easy to see how the performance compares over time. It's completely obvious when something isn't how it should be. And I can also get an idea from the previous plot, like when performance starts to drop, then I can preemptively arrange an instrument cleaning before performance drops below an acceptable level. And there are the other plots, which I will show a couple of them, that they can give indications as to why the performance has dropped on the machine. So looking at the problem that I had towards the end of August, I had to vent my instrument late at night because I had a problem with an O-ring, which actually cut one of the voltages to some of the source, and I had to take it out and reseat it. And I was doing this at about 10 o'clock at night, so I was baking out and calibrating the next day. And of course, as these things are, I was going on holiday. So I calibrated and started my QC run and hoped for the best. But actually, I'd probably been a little bit impatient myself. And so the mass accuracy had actually drifted from when I calibrated to when my QC runs were coming off the LC at the end. And it really wasn't possible to see this just looking at my MS1 chromatogram, right? Everything looked absolutely fine. The intensities were not a problem. But what I could see from the QuiC, looking in at the mass accuracy of the IRT peptides, was that actually, I hadn't waited for my Orbitrap temperature to stabilize. As the temperature sort of normalized over time, then the mass accuracy was drifting a little bit. And what it meant was that, fortunately, my lovely technician came in the following day, recalibrated the machine, and then everything was fine. And then I had the same problem actually a few weeks later, not me this time, but with another thermo engineer, also a bit of a speedy calibration, and we see the same phenomenon. So yeah, and another thing that I've used it for, and this is very recent, so not long after we had our QuiHFX installed, the performance dropped really, really rapidly. And as I'll show you in the following graphics, you'll see that the DDA mode suffered much, much worse than the DIA. And looking into things a little bit more closely, this allowed between myself and Thermo to diagnose a problem with the quadrupole transmission, which was not possible to see from anything on the tune page with the CalMix. All the calibrations were passing, and we had to arrange then for a full instrument cleaning, as it turned out that actually there was some oil inside the instrument. So you see here the same plots before. This is now of my QuiHFX, and you see performance started to drop almost as soon as I'd run my sort of 5 replicates of my QC right at the beginning of the installation. Then performance just started to drop, and in DDA it just dropped like a stone very, very quickly. But what you see with the DIA is that performance dropped, but nowhere near to such a dramatic extent. And this was really what helped us pinpoint the problem with the quadrupole, because of course in DIA one is using much wider windows than you have in DDA, and so it doesn't suffer as much in terms of the transmission losses as when one is trying to isolate a 1.6 Dalton window in DDA. So this meant we could really go straight for the quad when the engineer came and go for the cleaning. And it was fortunately, as you see from the right-hand side of this plot, performance now seems to be up and stable. Actually, now I move on to some other things that I've also been using QuiC for, probably not necessarily what it was designed for, but I also find it really useful. So when I got my new QE, I was also messing around a little bit with what could I get out of it in terms of limiting amounts of material. So we do a lot of work on stem cells here where we don't get very much material, and trying to optimize the run times for particular loads of protein and everything else. So I was playing around a little bit with different lengths of gradient and loading different amounts of my QC sample to see where I get to. And what's really nice about QuiC is you just have this such an easy visualization, right? So with the 90-minute gradient, you know, it's clear that every time I load a bit more, I gain something in terms of the number of precursors. What's interesting then, if I go to a shorter gradient, is that the number of precursors I get from the 100 nanogram load actually goes up compared to the 90 minutes. So there's always a trade-off then between, yeah, the runtime, obviously getting as much information, but also the peak heights. When you compress the peaks a little bit more in the shorter runs, you can actually gain a little bit more information. So this has been really useful for us to inform about the run times that we would choose when we're really working in these sample limiting amounts of material. The other thing I use it for is also, again, more method development. In terms of the DIA now, the really nice thing about QuiC is you can just look at all the different kinds of data about how many different segments in the MS2 part of the DIA gives optimum performance. So this is now on my long gradient, and just a couple of replicas to show that it was at least reproducible and not a sort of one event wonder. I just zoomed in a little bit of my my plot so that you can see what's going on, and that it's clear with this instrument. It's not necessarily the same across all instruments, so that's why it's really nice that you've got these instrument-specific views. Is that I gained something by increasing the number of my DIA segments on my QE in both the number of precursors that I get, and also you see it reflected, obviously not such a big shift, but still reproducibly more, that I get more protein groups out of having more segments in my DIA. So yeah, in order to summarize, because there's the built-in search engine, I can make the custom QC criteria from my sample against which to measure my instrument performance, and I can have this for different instruments for different runtimes. I hope in the future, when I get my new computer, that I'm going to put this on, that will mean that everyone has access to the QuiC history. It should enable us with multiple users to easily identify any loss in instrument performance with regular checks of this QC sample. I actually just learned that it's possible to tell QuiC to monitor a QC data folder on the server, so actually then even less effort will be required on the part of the user to keep an eye what's going on, because it will upload the data automatically when it sees it in a particular folder. And yeah, I really like the powerful graphics because they let you monitor a whole number of different instrument parameters, and that can really help you diagnose where there's a problem, when the performance goes down, and yeah, this has meant that at least I get a quicker response and a solution from the instrument vendors and less downtime on my machine. It's also actually a really quick and easy way to visualize how the changes you've made in method development experiments affect the outcome, and it's really fast and easy then to pick the optimum conditions depending on sample load and gradient length, for example, in both DDA and DIA. So with that, I like to acknowledge everyone that I work with closely here at the Institute, so the group of Alessandro Ori, the other people in my core facility, also the core facility bioinformatics who helped me out a great deal. And I provide a link to our website and my contact email address in case anyone has questions for me after this webinar today, then feel free to drop me a line. Okay, so yeah, I'm done, Florian.
Good. Thanks, Joanna, for this excellent presentation. I think with this, we are actually almost at the end of the webinar. We're now staying in a couple of more minutes. You can actually ask us questions using the questions form in your browser. And then either Joanna, Ian Leonard, the main developer of QuiC, or Sebastian Müller, R&D scientist diagnosis, will actually be with me to answer all of your questions. So go ahead, let them coming to us. Okay, the first question that I've got is there any way to measure the carryover from the QC sample? So what I can actually briefly tell you how we do it at Biognosis. We are what we are using is our QC sample is C. elegans, and we take this sample because it's something that we hardly ever measure to actually be sure whenever we get carryover so that we can clearly identify them. Then the next question is, and I think that's more addressed to you, Joanna, is how often is the QC sample injected?
So I run my QC sample at least once a week, of course, from my side. Every time I calibrate, I want to check that the calibration was the same as the performance before. And then the QC samples also run at the beginning and the end of every batch, and quite often in the middle of very long batches of samples. So some days we have experiments that take days of instrument time, and of course then the QC batch is inserted, the QC sample is inserted into the middle of these longer batches of sample to make sure that we can really ensure that everything's running. So I would say that a QC sample runs on average at least once every 24 hours.
Good. And the next question is about the cost of QuiC. So QuiC is free of charge, so it can be downloaded directly from our online shop, and only needs the IRT peptides or the HRM peptides to be functional. And the next question is about the monitoring of the retention time drift. So yes, QuiC can both give you indicative plots of retention time drift, and you can actually monitor that both in the IRT dimension, but also in the retention time dimension. And as shown in some of the early plots, actually you can even show the LC stability within one run. If you've missed the beginning of the webinar, after the webinar we'll actually send out a recording to all registered people. Then another question is about the handling of corrupted files. So one of our guests wants to know how this is possible. Ian, can you please comment on that?
Yep, this should be clarified that currently this is supported only for thermo formats. What QuiC uses is we wrote our own small applet, which opens up the runs and iterates through all the spectra and makes sure that everything makes sense. And if that applet crashes, then the main application will handle that issue itself and inform the user of that. So.
Good. Then the next two questions, or the next three questions, are again for Joanna. First, from one of our guests, They want to know what column length do you use for the QC runs, and why do you use longer gradients for DIA and shorter for DDA?
So I actually do all of my QC on a regular basis with the long gradients. That's sort of my longest gradient. My column length is 25 centimeters. And the reason why, like, the inter-batch and intra-batch QCs are 1 hour, I didn't show any of the folders for that, but I have a separate folder for those. That's just to, for these inter-batch QC runs, that they're a little bit shorter. They don't take so much of the instrument time. And it often also depends on the length of time that the user is running their samples. So that's, of course, dependent on the sample itself and the complexity. So my setup is probably a little bit more complicated than I showed you. I tried to make it a little bit simpler, but yeah, that's the general idea, that it's yeah, regularly with the shorter runs, the longer runs at least once a week, and if people are running the longer runs as part of their batch, then also there as well. Yeah, I hope that makes a bit more sense. And I'm also just working on, like, yeah, especially with the QE, with these different gradient length times, trying to make dedicated performance criteria for the different run times, so people can sort of selectively take the one that matches what they ran with their real samples.
Good. And then there's another question about how often do you actually calibrate both the Lumos and the HFX, and also how often you have to clean them actually.
So I can't really comment on the QE because it got oil in it on the installation, so it probably needed cleaning much sooner than it otherwise should have done. I calibrate both instruments once a week. Of course, the QE has some inbuilt time from thermo that says you should do it every 24 hours. I mean, this just doesn't make sense in a proteomics facility where you're running these nano columns, and batches of samples really run over days. But I do keep an eye on, obviously, the mass accuracy drift over time. And before I decided that I could manage once a week, I was doing it, looking how the mass accuracy drifted over time. And I have no issues with the stability of the mass calibration on my machine if I calibrate it once a week. Thermo say 24 hours by default. They might recommend you change it to every 3 days. I mean, it really depends on how you're running your instrument. Like I said, I sometimes have batches of samples which I run over many days, and it just wouldn't make sense to swap sources and things and calibrate in the middle of a batch of samples. So, and I find I don't need to do this. The Lumos is really stable for once a week, and the cleaning at the moment, I've been monitoring obviously the Lumos for much longer, and I clean my instrument every 3 months, so quarterly. And by doing that, at least I've not really suffered any major losses in instrument sensitivity over time. But I'm using, 15 years of experience to know that maybe if I pushed it a bit further, I might start suffering. So I rather preemptively arrange for it to be cleaned than wait for it to go down. Yeah.
Good. The next question is about the QuiC installation. So generally, you can actually install QuiC on your local instrument computers, and there actually the automated corruption checking comes in really handy, because then you're sure that the software will actually not clock your computer and then lead to a crash of your mass spec. But in general, you can also install it on any other computer, and with the folder monitoring, you can then actually automatically pull out the data from the folder and analyze it as Joanna described. And then one person asked about QuiC and if you can extract information about pQuif. So in QuiC, we are actually having a plot about the full width half maximum of the IRT peptides that is displayed within the software directly. The next question is about if QuiC can actually automatically terminate the measurement queue if there is a drop in any of the numbers. If you might comment a little bit more on future plans of QuiC, because that's something that's currently not possible. So currently QuiC does actually not communicate with the instrument software or the sequence queue. But what we're also trying to implement in the future is more of a warning messaging system, and I think Ian might be able to comment more a bit about that.
Yeah, so what's planned in the future is for the user to actually set their own thresholds, hopefully within the plot, so they can see that if the plot falls below a particular threshold, that the user will at least be notified of these issues. But this always depends on the instrument, so we leave that to the user to decide those values.
Good. If there are no more questions, I'd actually say thank you first to Joanna for presenting this very exciting work about how she uses QuiC to monitor the performance in her facility. Then I'd also like to thank you all out there for listening to us today in this webinar. This was the last webinar of diagnosis for 2017, but be sure we'll be back in 2018. And with that, I wish you all a nice day and a nice holiday season, and goodbye.